last evening, I spent most of the time trying to give you some tools and analytic tools and a way of thinking about ethics, and I gave some examples of certain interventions. Today, I am going to provide a couple more tools, but mainly it's going to be looking at interventions and trying to figure out what's going on with them. But I ask again for us to pray for help. Gracious Father in heaven, I am asking you again to help me speak clearly and correctly. Let me not steer anyone in the wrong direction on these weighty matters. Let me only speak truth driven by a serious kind of love. For these, my neighbors, my brothers and sisters in Christ, as I reveal what is really happening in modern medicine. With these high-stakes life and death issues, I know that for those facing these heart-wrenching dilemmas, faith in you can be shipwrecked. For these, please let truth point to an infinitely greater hope than what the world can offer. And I ask this in Jesus' name. Amen. This morning I'm going to continue on what we started in yesterday's last lecture, that is in vitro fertilization, by assessing other developments which would not have been possible without IVF, such as embryo donation, embryonic stem cell research, cloning, preimplantation genetic diagnosis, three-parent embryos, and gene editing. Now, as we discussed, in vitro fertilization was originally developed to treat infertility, but in its essence, it's not solely and specifically for infertility. It's the process of conceiving a child outside the body. Well, actually, to be more precise in definition, it's more general than that. It's the process of conceiving mammalian progeny outside the body. For the purposes of this class, it'll be implicit that I'm discussing issues related to humans unless I specify otherwise. Realize this does make a big difference in ethics. It's that imago Dei thing we discussed yesterday. So killing an innocent pig and eating it may be perfectly ethical. In fact, when the belly is prepared as bacon, I highly recommend it. Doing that same process to a human being is not so ethical. To understand embryo donation, I need to present more of how IVF is typically practiced. Conceiving more children than the parents plan to raise, which is problematic in and of itself, but also leads to further immoral practices. Before I dive into those practices, though, I wanted to point out a few assumptions that I use in this analysis. The logic and arguments for these assumptions are beyond the scope of this presentation, but I think you'll find them pretty uncontroversial and straightforward. The first assumption is that parents have a moral obligation to raise their children. The minimal requirement of this is to make every effort to provide at least a suitable environment for the children to grow and develop. This would mean, at the minimum, parents have a moral obligation to provide hydration, nourishment, and shelter from exposure for their children. Although it is controversial as to what upper age or advanced level of development this obligation ends, for instance, are you supposed to cut off your children at 18 years of age, or 21, or 30? So although there are differing opinions as to what upper age this obligation ends, all would be in agreement that it would be certainly unethical for parents to intentionally abandon a child at the, say, infant stage of development. No one would argue with that, or for that matter, abandon an even more dependent child at some earlier stage of life, such as before infancy. Abandoning such a young child would be equivalent to the ancient practice of exposure, a form of infanticide. In ancient Rome, because ultrasound had not been invented yet, you waited for birth to check the gender of your child. If she's a girl and you didn't want a girl, you exposed her to the elements, meaning she was tossed outside to the city gates where the dogs would come and eat her. That's one example of exposure, and it's unethical for parents to do such a thing. For the purpose of discussing IVF, I will limit this assumption to children at these earliest stages of life when they are totally dependent on their parents to survive, meaning I'm not talking about when to send your kids off to adulthood. As implied earlier, the negative way to state this first assumption is parents should not abandon their young children. Now, there is a hierarchy of morality with abandonment. Abandoning a child with no hope for rescue, for instance, leaving an infant exposed in a remote forest, is the equivalent of murder. Abandoning a child with some hope of rescue, for instance, leaving a child where a passerby might rescue the child, is not as immoral as murder, but it is certainly not commendable. Relinquishing a child to an alternate situation, for instance, an adoption center, where she may be adopted and raised, is possibly morally commendable only if the parents are in such dire straits that they cannot provide the minimal environment for the infant to survive. Certainly, an upper middle class American married couple should not be commended for relinquishing their infant child to an adoption center, simply because they decide that her care and feeding is not a priority for the household budget. The second assumption is that conceiving a child outside the context of a heterosexual marriage is unethical, such that when analyzing IVF, I will assume and restrict my comments to IVF done for a married couple, one man, one woman, using their own respective sperm and ovum. Now, one more concept I need to bring up is something I call strategic euphemisms. It's a strategy of using words to confuse, deceive, or otherwise cover up something unpleasant or immoral. I try to use terms to clarify and unwind these euphemisms. The terms I use may sound odd because the euphemisms are the standard terms used by not only pro-murder writers, pro-choice writers, but distressingly accepted by pro-life authors as well. The phrase I used, more children than the parents plan to raise, would commonly be stated in the IVF world, more embryos than the couple plan to use. However, I'd like to point out that as soon as offspring are created, the couple involved in relation to its offspring is by definition a parental pair, commonly called parents. Parents do not typically refer to their offspring by their stage of development, such as embryo, but by their relationship to their offspring. For instance, I have two daughters. When my older one was 8 years old, I did not introduce her as my prepubertal. And when she is 30, I will not say as a greeting, "Allow me to introduce you to my adult." No, I have always referred to her as my child or my daughter, not by her stage of development. Regarding the term use, parents do not use children. They raise children. Exploiters, such as pimps and human traffickers, use children. Let's see. Oops. Sorry about that. These strategic euphemisms are inaccurate and cloud the ethical picture by dehumanizing children at their youngest, most vulnerable state, and consequently they portray them as objects with no moral standing to be used instead of raised. They're discarded, meaning exposed, or they're donated, but in reality abandoned, or they're destroyed, a euphemism for murdered. So I'm very careful about the terms I use for the sake of accuracy and clarity, given all the euphemisms and deception. Let's see here. Sorry, had a slide advancement issue. Okay, yesterday I presented two concepts which I categorize in fallacies and deception. Strategic euphemisms fall under this category as well. Now, going back to this issue in IVF of parents conceiving more children than they intend to raise, because of the expense, discomfort, and risks of egg collection and the relatively low probability of a single embryo child to survive to birth, the highest published success rate for a live birth is 55%, and this is with 2 to 3 embryos transferred in a single IVF cycle. Multiple eggs are typically harvested and multiple children conceived by IVF. 2 to 3 of these children are then transferred into the mother's hormonally prepared uterus. The rest are frozen or cryopreserved and stored. These children are sequentially thawed for further cycles until the parents have brought to live birth the number of children they desire to raise. Any remaining cryopreserved children unwanted by their parents will have one of these 3 dispositions. One is to have them killed. There are different ways this killing is typically done, but ethically, they're all effectively the same: intentional killing of innocent children. One method is to have the IVF lab personnel thaw and discard the children. The parents can direct the lab to do this immediately, or they can do this passively by initially paying the storage fees to keep the children cryopreserved, but then quietly ceasing the payments. In the passive method, they do not make contact with the lab, so they are not actively directing the lab, but without the storage fees paid, the lab cannot afford to cover the storage expenses in perpetuity, so one option is to discard the cryopreserved children. This is no different than the ancient pagan practice of exposing children to the elements in a place where there is no hope of rescue, such as a remote field. The children will surely die. That is the intent of the perpetrators, both the parents and the lab personnel. The tool employed is the hostile environment. The other method of killing the cryopreserved children is typically called, quote, donate to research. This is a particularly heinous euphemism, as it not only dehumanizes the vulnerable children, because after all, objects are donated, people are not, but also implies goodness. The term donation implies a sacrificial good. That term is being used for what is in reality wickedness. Parents should not be commended for relinquishing their children to a researcher who openly intends to experiment on and kill them. So one disposition option for the remaining cryopreserved children is to kill them, which is unethical. A second possible disposition option is to abandon them to a situation where another couple may rescue them and adopt them. This option has been described as, quote, relinquish the embryos to another couple for reproduction, end quote, using the phrase, quote, donating to another couple, end quote, to describe what the parents do with their children. Once again, the euphemism donating muddies the reality of the humanity of the children. Furthermore, the term reproduction is wholly inaccurate to describe what the adopting couple intends. Reproduction has already occurred because the embryos are the children of the couple who conceived them through IVF. The adopting couple is not being gifted some remarkable material to help them in reproduction. They are rescuing children who really and not just potentially exist and would otherwise perish. The adopting couple is not reproducing. Reproduction has already occurred, as I point out. Though this abandonment option is not as evil as directly killing one's children, it is certainly not commendable. Now, there are circumstances where this may be morally acceptable, but not in the context of children conceived through IVF. For instance, when a single impoverished teenage girl has an unintended pregnancy and the father of the child has abandoned that teenager, it may be morally acceptable for her to relinquish her child after birth to an adoption agency. The circumstances for the parents in the IVF situation the circumstances are entirely different. First of all, they make up an intact married couple. Secondly, they are not impoverished. They have means. Only people of means can afford the tens of thousands of dollars per cycle for IVF. And that's cash. It's not covered by insurance. Third, they are relinquishing their children to a situation where if they are not adopted, and there is certainly no guarantee, the children will die. This is in contrast to a child given up after birth. If the child is not adopted, she will in all likelihood survive to adulthood and independence. Finally, the fourth difference. The conceptions of the IVF children were manifestly and by definition intended. Not only not an unintended pregnancy, these children are about the most intended you can possibly get. So to recap, one disposition option for IVF conceived, unwanted, cryopreserved children is to kill them. Another option is to abandon them. Not as immoral as killing them, but still unethical. The final disposition option for cryopreserved children is for their parents to change their minds and raise their children instead of killing or abandoning them. This would be by thawing them, then transferring them into their mother's hormonally prepared uterus. This is the only ethical disposition option for parents of cryopreserved IVF conceived children. Certainly, if parents choose the unethical option of abandoning them to an adoption center, then it would not only be ethical, but commendable for another couple to rescue the children by adopting them. Distressingly, there are Christian ethicists who have unwittingly fallen for the strategic euphemism, and by doing so, equate so-called embryo donation with embryo adoption, making the illogical conclusion that because adoption is ethical, then embryo donation must then be ethical. This is unintelligible because whether one uses the term donate or abandon, a donor is doing something entirely different than a recipient. The abandoning parents are doing something entirely different than the adopting parents. Moreover, it would be absurd to designate an immoral act, in this case abandonment. It would be absurd to designate that as ethical on account of the virtue of the rescue operation, in this case adoption. The rescue operation that was necessitated by that very act initially. We talked about it briefly in the Q&A session. I'm going to give another illustration here. So this illustration, to clarify this fallacy again, is this fallacy which is distressingly pervasive. Some of you may recall that back in 2013, the Westgate Mall, an upscale shopping mall in Nairobi, Kenya in East Africa, was attacked by terrorists who killed many civilians and kidnapped many as well. There were some courageous first responders who were outgunned but were able to rescue many in the mall. It would be absurd to call the terrorist attack ethical on account of the virtue of the rescue operations necessitated by that terrorist act. The terrorist act and the rescue operations are not the same and should not be equated. The virtue and courage of the rescuers do not confer virtue upon the terrorists. That would be absurd. Such is the parallels with so-called embryo donation and embryo adoption. We've already discussed so-called embryo donation, which is really child abandonment. The next one is embryonic stem cell research and potential uses, or so-called therapies. Here's a bit of the science to understand this. So you have, let's see here. So this is a little description of, you can see that, little description of a cartoon of fertilization occurring. So you have a sperm cell, you have the egg cell, and then you now have a conceived child. That cell divides into multiple other cells that are all the same. Now at this stage, they're called totipotent cells because, well, they're totipotent because we can't tell the scientists can't tell the difference as to which cells are going to become what. So the assumption is that all those cells could become anything. But let me make a point that it just means that we don't know which cells are going to be turning into what. It doesn't mean that it's random. So these cells then eventually differentiate into something that we can tell difference from about 3 to 5 days later, called that we call the blastocyst stage of development. And so we have this outer cell mass that we call the outer cell mass, and then the inner cell mass. And interestingly, isn't this interesting how this? I pulled this illustration from the web, but now it becomes a human fetus. So before it's, I guess, a blastocyst that might turn into a frog, or a morula that, I don't know, might be a gerbil. But now we can see that it's going to be a human. Let me just make the point. It's a human blastocyst, a human morula, a human oocyte. At any rate, the outer cell mass is eventually becomes the placenta and the parts that the child is using to survive inside the womb. And then the rest of the child, most of the child, is the inner cell mass. So with embryonic the inner cell mass cells will go on to become the rest of the child, differentiate into different organs and things like that. So at this point, they're considered pluripotent cells, meaning they can change into all sorts of things. So the thinking is, well, we can pluck out some of these cells. Maybe we can tease them and differentiate them into certain types of organ tissue. And let's say you have a failing heart, we can replace that with these stem cells. Maybe you have a failing brain, we can change them into brain cells and stick them in there. Maybe there's immune system stuff, and isn't it wonderful? The problem, of course, is that when you pluck those little cells out, we kind of need them to grow up and develop, you kill the child. So that's a problem. There are hopes with this for regenerative medicine, such as spinal cord injuries, to replace spinal cord cells. Just within the past, so up until recently, there really has not been any successful therapies that have been derived from this. But just within the past year, there is an experimental treatment for macular degeneration, a very common cause of blindness in older adults, using embryonic stem cells that has shown some success. And this is distressing because they consider it in, there's different phases of clinical trials in terms of bringing a therapy to market. So in the West, in the US, you have something called preclinical trials, which is done so you test on animals. Once you see that it's fairly safe and all that stuff, then you might go on to phase 1 trials, which is experimentation on humans. But you would do it in very grave situations or things like that. And then eventually you get to 2, 3, 4, phase 4, and then it's completely released. So it's already gone through what they call preclinical trials, because the animals that they use to test it on are they're stem cells. They're human stem cells. In other words, they're calling these children not human because they're preclinical trials. So that's the way you get around it. It would be as if you just, take a 2-year-old child and experiment on them and just say, well, this is preclinical trials because I designated them as not human. Okay, so the ethics of it. To analyze the ethics of this, we look through the principles and concepts that we now have at our disposal. And this is all the stuff that you guys learned last night. From biology, we know that the blastocyst is actually a human being in the blastocyst stage of development, so is a being created in the image of God. Was there a little spin that happened there? Okay. Biblical commands on image bearers. So we know that the biblical commands on image bearers apply here. We note that human beings at that age are incapable of committing a capital crime, so by definition is an innocent human being. Now, killing that innocent human being is murder, which is prohibited by specific biblical command. Therefore, killing a human at the blastocyst stage of development in order to get her stem cells is unethical. So that's pretty straightforward. Excuse me. Now, there is a category of human stem cell research that can be not only not unethical, but can be very commendable, and that is adult stem cell research. There are stem cells found in fat tissue that have been manipulated to provide therapy to patients. Adult in this case just means that these cells are found in humans who have grown to the point of having unipotent or differentiated cells, meaning anyone beyond an embryo stage of development. Usually these cells are taken from some age after birth in order to treat that same person from which the cells were taken. So it's no different ethically from doing, say, a skin graft. In fact, one of the reasons why it works is because you don't have the problem of immune rejection. So this adjective, embryonic, is very important when figuring out whether an intervention is ethical or not. Like I said earlier, it's pretty straightforward that human embryonic stem cell research is immoral. But what will we do if and when an effective therapy for a life-threatening or even, oh, thank you. But what will happen if we get an effective therapy for a life-threatening or even a non-life-threatening disease that gets developed using human embryonic stem cells? Let's go back to the principle slide. In this case, the problem here is complicity or cooperation with evil. So even if you're starving to death because you're in Samaria under siege by the Syrians, and even if you didn't slaughter your roommate's child, and even if you didn't cook the child in the pot, when you ate the child's flesh to save yourself from starvation, you are complicit with the child's murder. So I think you may be familiar with this. It's from 2 Kings chapter 6, verse 28, regarding the siege of Samaria by the Syrians at the time of Elisha the prophet. Starving mothers eating their own children, and really not seeing any problem with it. The only problem that came up to the king was, well, wait a minute, we were supposed to kill her kid today, and we didn't get to. This is unfair. It is astonishing that there wasn't an issue of, maybe it wasn't right to boil that child and eat him. Okay, so the fact that most scientists think it's totally ethical to kill the vulnerable to benefit from their body parts is nothing new or anything particularly progressive in thinking. It's as old as dirt. It's called might makes right, meaning if I'm bigger than you, then I have the right to do anything to you or take anything from you. So really not that, like I said, not that new. So now cloning. The kind of scientific term for human cloning is something called somatic cell nuclear transfer. Somatic cells are, we categorize two types of cells in the body. Gametes, which are the egg and sperm, so those are the cells that will go on to reproduce. And then the somatic cells are everything else. And the difference is that the somatic cells have a full complement of chromosomes. So every cell in our body, except the gametes, have 46 chromosomes, 23 pairs. The gametes have 23 chromosomes, so it has half the number. So let's see if I can get this here. So how does cloning work? Well, you take the somatic body cell, you could take a skin cell or any cell from your body, and you pluck that out. You take the nucleus from it. I'm sorry, let me take another step. So you got a somatic cell, and then you have an egg cell, a gamete. So not a sperm, but an egg. The you take this egg cell, and then you pluck out the nucleus that is, the nucleus of the egg cell. So now you have everything except the nucleus. So it seemed like half the part. So you don't have the chromosomes of that donated egg cell. Then you take one of your skin cells, and then you pluck the nucleus out of that. So you take the 46 chromosomes out of that, and then you stuff it in the egg cell, and now you have a new human being. Scientists would say that you don't have a human being yet, but you actually do. And then and that human being grows, and now they say that it's a clone. So the scientists that are doing this type of stuff say that there's two things you can do with this clone. You can either do reproductive cloning, meaning you take that clump of cells and then you pop it in a surrogate mother and raise that to term. But recall, reproduction has already occurred. So this is a strategic euphemism, right? You guys got that. The other option that they talk about is therapeutic cloning. So you take that clump of cells, you and you take them as stem cells, or actually you can raise them up a little bit longer. I suppose you could do that too. Maybe you can raise them until they are 10 years old or something. You can take their kidneys out. There's really no reason not to. There's no ethical distinction from doing that as you would at the embryo stage of development. Anyway, you do a tissue culture and then you fix yourself. But notice what's going on here. So this is what's happening with human cloning. Let's see. So the ethics analysis. Well, it's similar to the reasoning around the immorality of IVF. What happens if you don't do this? Well, nothing. This is the inaction alternative. You don't die, you don't become paralyzed, nothing. In this case, short-circuiting divine design this has much greater uncertainty of damage to this child that's conceived. So you think that so if you think that we all came from random mutations that led us here, then you can think, well, we can do better than random because we're really smart. Except that if I came from randomness, how smart could I possibly be? But anyway, let's put that aside for a second. If we came from random, then, we know what's going on here. We can just do a switcheroo and it should be fine. But what we're discovering, even the scientists who don't believe in God, is that things are far more complex than they ever thought. But at any rate, so now you're going to do the switcheroo where you're going to pull the genetic material out of this, you're going to switch it around and all that. Do you think there's something else in this egg cell besides the nucleus that might have some impact on the child that's conceived? This is far more dangerous to the child that you're going to be conceiving. Let's see here. So that's the ethics of reproductive cloning. Similar to IVF, really not a good idea, but actually worse. As far as therapeutic cloning, same issues as with embryonic stem cell research, of course. They're made in the image of God, so these are children made in the image of God, and they haven't committed any crimes, so you're not supposed to kill them. So a human being is not to be used for her body parts then killed. So once again, unethical. Preimplantation genetic diagnosis. So in vitro, so preimplantation genetic diagnosis is this idea that you can sample some cells from a child in the embryo stage of development, figure out whatever it is that you want to figure out or think that you can figure out. For instance, whether they have a particular genetic disorder that's recognized, whether they're a boy or a girl, et cetera, and some days people would like to say whether they have blue eyes or brown eyes or, all sorts of things. Sounds really simple. So what goes on here? Remember this cartoon, so you have the fertilization that occurs, you have the child, you get to this stage, you get to that stage. So now what you do is once you get to the blastocyst stage, in this case, you actually are thinking that you want to raise some child. Let's say you decide, I want to execute my child that has Down syndrome. I know it sounds a little rough there, but that's really what's happening. What would you say if we did this to a 6-year-old with Down syndrome? All right, so you have this outer cell mass and you have this inner cell mass. You pluck out one of these outer cell mass cells because they'll have the full complement of the chromosomes, but it's not going to immediately kill the child in whole, because that's one that's going to become the placenta and stuff. And then you do an analysis of it. If the child has an extra chromosome that you don't like, then you execute them. And then you move on to the next child until you have one that doesn't. Or you want a boy, or you want a girl, then you can figure that out pretty easily as well. Now, let's see here. Another thing I should mention, I didn't mention about IVF, is when the embryologist, so the obstetrician gynecologist who does the IVF technique works with an embryologist who does the actual fertilization and all that. The embryologist then takes guesses at which children appear to be weaker or stronger. There's not a lot of science behind this yet, and they kill the weak and they raise the strong. So that's how it works. They look at these little babies as these little clumps of cells and saying, I think this is one that has the best shot. This PGD is kind of the next step beyond that, where you can now look at some other stuff and say, yeah, this definitely is the weakling. I'm killing him. So what's the problem here with preimplantation genetic diagnosis? Well, first of all, you can only do this if you do in vitro fertilization. So if you have a conventional God's design child conceived inside the uterus, you can't pop them out, pluck out the cells and all that stuff. So you got to do IVF, which we've already gone through. That whole thing is unethical. I do want to make a point, though, and I made the point last night during our question and answer time a bit too, but I want to be crystal clear again. When these children are conceived, even if they're in a test tube, even if they're going to be, genetically, analyzed and all that stuff, there now exists a child, and that child is a creature made in the image of God. So biblical ethics apply to that child. Even though the child was conceived out of wickedness, that child is not anything less than a creature made in the image of God. In other words, is a human being. So, and I stated this before, but I want to emphasize it again. So whether a child is conceived through the embrace of a loving married couple or through the violence of rape, the child must now be treated like a human being or human person or creature made in the image of God. The way the child was conceived conserves no additional virtue or additional iniquity on that child. I mention this because I talked about how, Protestant, that we don't have a particular stand because we don't have a pope, we don't have Vatican, so we don't make policy statements. But I mention this because what I said earlier, Catholic philosophers got it right in terms of their conclusion that IVF is wrong, but I think the reasoning that IVF is wrong, but I think the reasoning that they got there isn't right. And it actually matters, and here's why. Here's one example. So there's a Vatican statement that actually prohibits rescuing the children that are in the embryo stage of development. They actually prohibit embryo adoption. So they're totally okay with saying, okay, you did IVF, that's bad, but these children are frozen. Hey, parents, you got to raise them. But if the parents say, forget it, I'm flushing them down the toilet, or I'm gonna donate them to an adoption center, the Vatican actually has a policy saying, nope, that's not right. There is this distorted spiritual but not biblical reasoning about it being wrong for a child to be placed in the uterus of someone other than her genetic mother. This was specified in the Vatican document Dignitatis Personae, the dignity of the person, published in 2008. But the practice of this actually occurred much earlier. So in 1996, in the United Kingdom, there were about over 3,000 frozen children murdered by exposure because of a law that had been put in place when IVF was getting started, that if an embryo is frozen for more than 5 years, it must be destroyed if there hasn't been any disposition option. The British Catholic leaders lamented this problem, so they said, this is exactly why IVF is a problem, and you cannot do this, you cannot do this. But then they said, but you can't adopt them off either. That's wrong. So it's better that they have In fact, the proponents said we should have a respectful funeral for these children. This is why it's critical to not only have the correct conclusions in ethics, but also the correct reasoning. Otherwise, you get all sorts of distortions and wrong reasoning. So, let's see, getting back to PGD. It's simply a diagnostic test. So it's really a matter of what you do with the information. I'm sorry, taking it further. So first of all, it's a problem because it's IVF. But once you go beyond that, suppose someday there is a way to do PGD somehow in an in vivo or normal or conventional way without having to do IVF. If it got to that point, then realize that it is simply a diagnostic test. And so it in and of itself is not ethical or not ethical. We talked about a hammer is not ethical or not ethical. It's how you use it. All right, go on to the next one. So next, so-called three-parent embryos. Now note that the term used to describe this is category. This is what you hear in the popular press. This is what you hear in actually the scientific press. That term, three-parent embryo. Note that the term used to describe this is categorically different than all the others. The others are nouns that are descriptive of actions, versus this one, which is a noun describing an actual human being. So no, we are not analyzing whether or not a certain class of children are ethical or not. We should focus our attention to the action, which is conception. As we just discussed, no matter how a child was conceived, she is no more or less human if conceived by intercourse between a husband and wife, or by IVF, or by cloning, or by this three-parent conception process. This is really important. Words persuade. Truthful or accurate words persuade truthfully and accurately. And of course, the opposite is true as well. All right, well, on to what this three-parent conception thing is all about. So here's another diagram. This is a problem that was thought to be a way to fix a genetic disorder that affects a part of the cell called the mitochondria. So the mitochondria is within every cell, and it's sort of the power plant of the cell. So it creates the chemical ATP, which is used by the cell to do all sorts of things. So there's a disease with this problem, and so there's problems with it. But the reason why this is potentially what's considered a cure for this is that I said earlier that the genetic material is all in the nucleus of the cells. That's not actually true. We used to think that was the case, but actually what scientists have found is the mitochondria actually have some DNA, something called mitochondrial DNA. It's included in all egg cells. So it's always passed on to children, but males don't pass this on to children, only females do. So if a woman has this mitochondrial disease, then they can pass it on to their children. So how does this three-parent conception work? So you have the egg cell of the main genetic mom. So this is the mom that has the mitochondrial DNA problem, the mitochondrial disease. And then you have a donor mom egg cell. So someone unrelated, or actually it could be related, I suppose. Someone that it's not the mom, it's not this mom's cell. This is now the donor mom egg. So once again, you pluck the donor mom's egg cell out. There's some mitochondria in that egg cell, and that mitochondria DNA doesn't have that genetic disorder. So now you have this shell. You pluck out the nucleus of the main genetic mom, leaving the mitochondrial DNA that is diseased, and then you pop it into the donor mom's egg. You take dad's sperm or whoever's, and then you do IVF, and then bingo, you have a child. Let's see here. Let's go to our toolbox. So this is just a modification of IVF. Ethics issue is similar. So you do a risk-benefit analysis. What happens if you don't do this? Well, nothing. The inaction alternative. You don't die. Not having your own genetic progeny is not a life-threatening problem. Furthermore, for a woman who has this mitochondrial disease and also believes she's called to have children, there is a commendable option: rescue an orphan by adoption. In other words, have non-genetic progeny. This perhaps may be God's loving design to point a couple to adopt, which perhaps they would not have ever even thought of had the wife not had this mitochondrial disease. That's just one possibility. But she's not going. It doesn't actually treat the mitochondrial disease. I hope you see that. It doesn't fix the mitochondrial disease. The problem that they're fixing is not having children that have the mitochondrial disease. So short-circuiting divine design is not indicated. In this case, it's even more riskier for the child who gets conceived, because it's unknown what negative effects from switching out these cell parts, just like I talked about with cloning, the unknown unknowns. And what do I mean by that? Unknown unknowns is this idea. We all think that whatever we know today is all that there is to know. But the fact of the matter is, there are probably things going on here that we don't know anything about. Like, I think when I was in medical school, and I mean, I'm old, but not that old, we didn't know anything about mitochondrial DNA. That was a new discovery. We didn't even think that such a thing existed. Prior to the 1950s and 1940s, we didn't know about DNA at all. It is possible that we finally gotten to the point where we know every possible thing, and there will be no new discoveries moving forward. That's certainly possible, but it's not very plausible. So I'm making the point that we have unknown unknowns here. We're completely uncertain to the damage that might be caused. All right. Actually, I should mention it now. So this idea that we can do these kind of switcheroos in this incredibly complex system is at best naive, but can be better explained. What I just described is something that I call is a severe case of chronological snobbery. So that's a term that I'm appropriating from C.S. Lewis. I think I'm switching around a little bit, but it's a similar idea that I just talked about. And we always look back and we say 100 years ago, I can't believe how stupid those doctors were. They knew nothing. I can't believe the crazy things they were doing. Is it possible that 100 years from now, they will look back on what we're doing and saying, boy, what idiots, they didn't even know that existed? I think it's possible. It's at least plausible. Don't you guys think? So, I mean, of course, I'm being sarcastic, but in all reality, this is just, and it's, the best scientists don't think that way, but even great scientists kind of make these assumptions until a new discovery is found. All right. And then the final point of this is complicity, of course, because you're cooperating with the evil, since child murdering research continues with this. All right, the next biotechnology we'll now analyze is gene editing through a novel approach called clustered regularly interspaced short palindromic repeats, which I think many of you have heard of as CRISPR, or CRISPR-Cas9. A palindrome, as you may recall, is a word or phrase or sequence that reads the same backward as forward. So this is an example of a palindrome. Todd erases a red dot. This is an ironic one, ibophobia, because it means fear of palindromes. Okay, so it's not a real word yet, but it is a humorous one. You get the idea. So this CRISPR, by the way, no one invented CRISPR. All they did was they recognized it. That's another kind of interesting thing about science and scientists. So we see stuff incredibly complex, and we say, therefore I'm great. I'm a great scientist because I saw this. It's kind of like, maybe God is actually kind of the great one because he actually made it. All you did was you looked at it and you saw it. But again, if you think that this all came by randomness, then you're the great one to see randomness. But then it brings up this point about something so complex, then randomness is your God. That's your idol, because that's who you think made this. Isn't randomness wonderful? So going through this, again, scientists didn't make this, they just observed it. Now, what did they see? This is fascinating. I know this is a crazy-looking diagram, but I'm going to point to it real quickly. So what they saw was this CRISPR-Cas complex was a way for bacteria, apparently, to protect against viruses. So as if it were not random, as if there was some purpose behind it. So as some of you may have heard from, I think, Dawkins, one of the strongest atheists, said, biology is the science of looking at things that appear to have design but actually weren't designed. So let's just pretend like this was actually designed, like this bacterial cascade. This is amazing. This is just amazing. So you have a bacteria, you have this virus particle. So a virus isn't exactly alive, as we know, or dead, but it has material, and it does bad things to cells, living cells. So the virus attaches to the bacteria, sends their viral DNA fragments, sticks it into bacteria, does bad things, and harms them. But interestingly, there's this Cas protein that was discovered, and this Cas protein captures some of the viral DNA. It processes it, sticks it into a spacer, or creates what they call a novel spacer. So DNA are these, it's like a code, letters of alphabet, you can think of it. And it's sticking into these areas that has this, what they call a Crispr array. So that's that palindrome. It's that repeating segments going, and they're repeating segments back and forth, same way forward as backwards. So then it sticks this piece of the viral DNA into a place that's safe for the bacteria. So now it's not going to kill the bacteria, but it's going to be safe for the bacteria, that then transcribes it to RNA. So some of you have to reach back into high school biology or college biology, or maybe it's hearing it for the first time. It's just it's the way that we see cells transcribe this code. So anyway, so it turns it into this RNA, which then has this what's called a Cas2 protein, which takes those bits of RNA, processes them, and then sticks them onto this Cas cRNA, this RNA complex, so that the next time this virus comes along, this Cas protein will recognize that DNA from that virus, capture it, and then blow it to pieces. Ta-da, we're done. This is really quite phenomenal that randomness created all this complexity of design. So now, how are they using this? So the CRISPR-Cas technology, the scientists are using it to cut out certain pieces of DNA and then can repair it into others. So again, DNA is like a code, so I'll give you a little illustration. Suppose you were putting together a piece of IKEA furniture, and you came upon this sentence in the instructions: insert bolt D through washer J on top of hole panel B, something like that. But some of the words were smudged, so they're illegible. This is like a segment of DNA which is damaged. You can't get the furniture piece together properly without this segment. So what scientists can do is engineer the correct segment of DNA, then use CRISPR-Cas9 to cut the damaged segment out, then insert the correct segment in, which then results in a repaired segment. So that's, in a very simplistic way. Actually, what's going on with bacteria and stuff is far more complex than that, but that's essentially the deal. This defies all logical reasoning to think that this sophisticated defense mechanism came into being by random chance. For me, this was glorious to see the awesome wonders of God. This would be the least problematic of all the examples in the IVF-associated categories of intervention, because it could potentially offer wonderful treatments for genetic disorders. This doesn't have to be done to children in the embryo stage of development. The main problem right now is, and the connection to IVF, is that, again, children are being used as lab rats using CRISPR and stuff. I better stop now because I think I've gone over, so.